Good morning, everybody, and welcome to today's SPHN webinar. We will hear about the SPHN driver project Imagine today. And this project aims to build a Swiss-wide infrastructure for image-based biomarker research and analysis. I would also like to kindly welcome today's speakers, and we'll do a quick tour de table for you, a quick round of introduction. So the first speaker will be Matthias Guckenberger. Matthias is the chairman of the Department for Radiation-Oncology at the University Hospital of Zurich, and he is the main PI of the Imagine project. Then we will have Stephanie Tanadini-Lang. She's the project coordinator. We'll have Michael Bach from the University Hospital in Basel, Andrea Bink from the University Hospital in Zurich. Andrea is also a co-PI. Roland Wiest, he's professor at of Neuroimaging at the University of Bern and the Inselspital in Bern. Mauricio Reyes from the University of Bern, the ARTORG Center for Biomedical Engineering and Research, and Adrien Depeursinge from the HES-SO in Valais from the Institute of Information Systems. So, Matthias, I would like to give the floor to you, and we are curious about the progress of the Imagine project.
Thank you very much. First, I would like to thank the SPHN again for supporting our project, where we aim to implement radiomics as a component into the SPHN network. We apologize, but we want to make sure that we present ourselves as a network, as a team effort, and that's why we have distributed the presentation between a larger number of people than it may be usually done. So I will start giving an overview. If we talk about diseases in general or oncology more specifically, and we have three categories of information which are driving most decision makings in clinical practice. So for almost all solid cancers, we ask about the patient, him or herself, gender, age, and performance status being the most important prognostic factors. We need pathology, what kind of disease is it? And we need imaging, CT, MRI, PET imaging for staging of the disease. And these are the three major components for driving our decision-making process. How does that fit into the landscape of personalized medicine? If we look into the definitions of personalized health, personalized medicine, I picked two of them from the US National Cancer Institute, from the European Union. They have quite a focus on recognizing that diseases are more complex, that the diseases are more diverse, also in particular cancer, but they do have a focus on biology samples, meaning the person's genes and protein and environment. That's what is the driving force, obviously, in this definition of personalized health. And also, if you look into the definition of the European Union, it is spoken here of the individual's phenotypes and genotypes. Genotypes, molecular profiling, but also medical imaging and lifestyle data. So if you look into this definition, well, the idea was from the beginning to have a deeper understanding of the pathology of disease, doing genomic analysis. But what is important, how about the patient himself and about imaging? Shouldn't there also be a more comprehensive and quantitative information being available about the patient, him or herself, and in particular about this imaging which is being available to better characterize, to better select the right treatment for the right patient? If we talk about imaging, we know that there's a huge diversity being visible in these images. And this is just one representation, which is all the same in the radiological report. It would all say these are 3 patients having non-small cell lung cancer and locally advanced disease. But effectively, if you look at the images here, and if we look at the 3D rendering, they represent very different patients. So the growth pattern, how this cancer develops inside the host, is very different. And it is the hypothesis that this development of cancer inside a host environment is not just a purely coincidence of luck, but it is the representation of the interaction between the disease and its genetic and epigenetic background into a host environment. And this becomes even more obvious if we not only use single modality imaging, but if we use multimodality imaging, where we can illustrate and define different biological processes in each of these layers, and have even a more and more precise, more deep understanding of the disease phenotypes as it is representing on this imaging. And that's what radiomics is all about. Radiomics is a methodology of quantitative image analysis which starts from medical images, where we segment these images, and that could be CT or MR imaging. We use mathematical formulas to quantify what is being seen on these images, not just descriptive as it is done by radiologists, but quantitative. And then using this information as it is done in other omics analysis to use it as an outcome predictor. So a comprehensive mathematical and quantitative way of image analysis. This information coming from radiology and from imaging is, to our understanding, truly complementary to the approaches we have from other omics approaches. So we know that the genomic background of the patient, epigenetics, transcriptomes, and proteome, they're associated, strongly associated with outcome of the patient, and they are driving forces to select the best treatment for each and every individual patient. But how could radiology fit in here and radiomics? Well, it could fit in between here. So the growth pattern, as mentioned before here, brain tumor, will be influenced by the genome, by the epigenetics, but it will also be associated with outcome. So this is the benefit or an advantage we could have, kind of translating from the image forward to outcome and backward to the biology background of the disease. Another complementary aspect of imaging is that we do have both a high spatial and temporal resolution. If we would, for example, investigate a metastatic disease, you can only take a biopsy at a certain period of times, and you can only biopsy few lesions. Liquid biopsy, that could be done on a more highly repetitive pattern, but it will be a mixture of all the metastases, of all disease phenotypes being inside one particular patient. And the advantage of imaging is that you both keep a high temporal, but also have a spatial resolution for either illustrating intra-lesion or intra-patient heterogeneity. So what we see as a vision for personalized health, that we not only focus on the pathology for a deeper understanding, but that we also complement Swiss Personalized Health Network for a quantitative data based on imaging using radiomics, and also potentially, that is not our project, for a deeper understanding of the patient, a deeper characterization, for example, using the variables. So more than just genes. So this is the schematic illustration. We have a heterogeneous disease. We have multi-omics approaches already being implemented. We have patient and patient preferences. And we want to complement the Swiss Personalized Health Network with imaging and quantitative image analysis to have a better understanding of the diversity of diseases. And we want to use a glioblastoma as a use principle. This is an illustration what we try to achieve. We try to achieve a network and a framework to use quantitative image analysis in the form of radiomics and make it available to the Swiss National Health Network. That involves standardization processes for imaging, image curation and storage, analysis of data, and of course the integration process. We have selected glioblastoma as a use case, which for people who don't know it, it's a primary brain tumor, the most frequent one in adults, highly deadly disease where we truly need progress for our patients, and where we do have a very good rationale that imaging might contribute to a better understanding of diseases. This is one early study where it was possible to start from images, distinguish different clusters of growth pattern. From there, seeing that these growth patterns are associated with different prognosis, and that also leads to different genomic and biological pathways being activated in these different imaging clusters. That is the overarching goal. And with that, I would like to hand over to Dr. Stephanie Tanadini-Lang, who is a group leader for clinical and medical research at the University Hospital Zurich, and who is coordinator for that project.
Thank you. So maybe you can go to the next slide. So I will give you a short overview of the project. Let's start with the participating institutions. We have institutes from all over Switzerland. We have the University Hospital in Zurich, the University Hospital in Basel, the Inselspital Bern and the HES-SO, and the hospital in Ticino, who all contribute data to our project. And then we have a couple of institutes, the University of Zurich, the University Hospital of Zurich, the University of Basel and the University Hospital of Basel, the Inselspital Bern and the University of Bern, and as well as the Institute of Information Science from Valais, who all contribute to analyzing the data. So it's really a Swiss-wide network. If we go to the next slide, we will see the main contributors, the co-investigators of this project from the institutes I have just mentioned. Most of them will later talk to you in their work packages and will then be introduced to you. But let's go to the goals of the project. We have on the one hand the technical goal. The technical goal is really to build a Swiss-wide infrastructure for image collection, image analysis, and imaging biomarker research. But this should be connected to a clinical goal, and the clinical goal is the identification of prognostic imaging biomarkers for glioblastoma cancer. I will explain in the next slide. The clinical data we are looking for, so as already mentioned, the proof of concept is on glioblastoma cancer. We would like to model overall survival after primary diagnosis of a glioblastoma cancer as the primary endpoint. And if there is data available, we also will look into progression-free survival. The clinical data set collected, we started with a large data set to be collected, but then we looked into the different hospital and we had to come up with a smaller minimum data set to be collected. So we look for overall survival and progression-free survival and some patient and therapy characteristics like gender, age, MGMT status, Karnofsky's index. This will be done for glioblastoma cancer first, but it can be later expanded to other tumor entities. For the imaging data, we focus in the first step on MR images. We ideally would like to have 4 different sequences. That's a T1 with contrast agent, a T2 flare, and a diffusion-weighted imaging. We want to have imaging at the first diagnosis before any intervention is done. And from these images, per image modality, we define more than 1,000 imaging biomarkers. They are calculated. But of course, this is only like a proof of concept with MR images. Later, it can be easily expanded to other imaging modalities, such as PET imaging or MR imaging or CT imaging. Here, the overview of our project. We have 3 work packages. The work package 1 is on standardization and quality assurance. So we know that if we define imaging biomarkers, we need very standardized imaging. We can't standardize everything, so we also have a part there where we look how robust are our features, but the main part is really to standardize the treatment protocols and to do regular QA. Then we have a second work package, and the second work package is on the IT infrastructure and on the data collection, on the clinical data collection. So how do we get the data in the different hospitals? Which data do we want? And how do we get this data on the Biomed IT nodes to do the analysis on them? And the last work package is then on the image analysis. And it's not only the definition of biomarkers, but also the definition of the tumor first, then the calculation of these biomarkers. And then as a last step, artificial intelligence approach, a data analysis approach to analyze the data and correlate to the overall survival of the patients or to other clinical parameters. And we have set up the project that we immediately start with the first 2 work packages. So we start with the standardization of the data, but at the same time, we also collect data retrospectively. Of course, this retrospective data will not be standardized, but we would like to have this data to already define the imaging biomarkers, to implement the routines for data analysis. And then later, when all the protocols are standardized from work package 1, as soon as work package 1 is finished, we would like to also have a prospective data collection. This has a very large advantage because we can then compare our models with the retrospective data and the prospective data. We all always talk about standardization, and it's very important, but with this approach, we can later show the value of having a standardized data set. We can show how much better we can predict the outcome of the patients. So this is the IT approach. So all the hospitals which contribute data are responsible for getting the clinical data, the treatment data, and the images. So each hospital implements that on their side. This implementation might be slightly different because every hospital has, of course, a slightly different infrastructure. Also, the different hospitals are responsible for the de-identification of the data, and they send this data then to one of the Biomed IT nodes. So whatever node they are connected to, they send the data there, and this data is sent further to the Zurich node, to Leonard Med, and then all the data analysis will be done on Leonard Med. So we aim that we have a fully automated procedure there. The data comes in, it gets automatically delineated, the tumor, the imaging features are automatically calculated, and we have machine learning infrastructure implemented, which makes the modeling part then, and which makes a model each time a new data comes in. So all the data analysis is done on Biomed IT. With this short overview of the project, I would already like to hand over to the different work packages. And the first work package, the first part of the work package 1 will be presented by Michael Bach from Basel, and he will show us about quality control of MR scanners.
Before we talk about MRI and phantoms and quality assurance, I want to start with a simple example. Next slide, please. And if you want to test your camera, you can take a picture of a test object. In this case, the test object is just a poster on a wall. And on this poster, there are some dedicated structures to test image quality. For example, the test pattern in the middle of the poster is called Siemens star, and it is shown in more detail on the right side of the slide. With this pattern, the resolution of the camera can be tested. Towards the center of the star, the black and white stripes come closer and closer together, and so they merge to each other. And because of the finite resolution of the camera, the stripes build such a gray area in the middle of the star, so they can't be resolved anymore. And the bigger this gray area is, the lower the resolution. Thus, it is immediately visible that the image on the bottom right has a lower resolution than the image at the top right. This is one example how we can test the real resolution of a camera. And we test our MRI devices in a quite similar way. Next slide, please. A camera takes a picture of the surface of the object, but with MRI, we can really look inside the object and virtually slice through it. Consequently, our test object is no longer just a poster, but a so-called phantom. The phantom we use in this project is a cylinder with further three-dimensional test objects inside. These objects are made of acrylic glass and are surrounded by water. There could be ramps, grids, holes, and other structures. The important thing is that we exactly know how these structures look like, and so we can test how good our MRI can depict these structures. If we put the phantom into the MRI scanner instead of a patient, we get these test images on the right-hand side. Analogous to the camera resolution test shown before, we can now test several imaging parameters. In total, we have 8 tests. Next slide, please. Here an overview of the perform a test is shown, and similar to a blood test, you can see the actual value of the parameter and its allowed range. The first two tests quantify image distortions. Therefore, the length and the diameter of the phantom is measured in the acquired images and compared with the real dimension of the phantom. Furthermore, it is tested if the resolution you set at the scanner's console is really achieved in the measurement. And the same for the slice thickness and the slice position. The intensity uniformity is analyzed. This basically means if the image looks homogeneous or if there are bright spots in it. Then the ghosting artifact is quantified and the achieved contrast is tested. In order to perform all these tests in an efficient and objective way, we set up a web interface where we can upload the phantom images and you will get an automatically created report. Next slide, please. Here you can see this web interface, and first we upload the images, in this case in total 34. We start the upload and the images are completely automatically processed. After approximately 3 minutes, one can download the results as a PDF report. And this report shows at the first side an overview of the tests as shown before. And the details for each test results and how it was achieved are shown in the following pages. You can also have a look at the longitudinal development of all test parameters. Then you can see trends in time for each parameter. And you can also export all these results to an Excel file for further processing if needed. Okay, this is how we do phantom-based image quality assurance. And the next speaker is Andrea Bink from the University Hospital in Zurich.
Thank you very much for your kind introduction. I have the pleasure to present the results and development of our standardized image acquisition and image reconstruction protocol. Next slide. The data pipeline is shown here, and you can guess from the position of imaging protocol in this data pipeline that's the central one. It's very essential how the images are acquired and in which way they are acquired and which sequences you use. Next slide. Even without knowing anything about MR physics and protocols and details of sequence acquisition, you can guess from the left side of this slide that the different vendors have different ways to present their imaging protocols, and this differs very much from MR machine to MR machine, from 1.5 Tesla to 3 Tesla. So you get a bunch of different information and different sequences and different imaging results, but that's not what we want to have. We want to have comparable images with standardized methods, and this is because we know that all of us, we want to have a better database for post-processing, like here shown on the right side of the slide, for automatic AI segmentation. Next slide. So why and how image standardization? We want a reduction of measurable measurement variability, because you know, for example, that TR can result in significant changes in imaging contrast as they are changed. We want to have a reduction of variability of contrast enhancement. That means to stabilize the time between contrast agent injection and image acquisition. Furthermore, it is necessary for automatic volumetric segmentation and feature extraction that you have a high reproducibility. We want to have reliable assessments of treatment response, which is very important for our clinical partners. We want to have a better quantification of tumor radiomics. And of course, it is a nice side effect. We want to have a decrease of the needed size of training data sets for AI algorithms. Next slide, please. So how can we achieve our so-called consensus best of Switzerland brain MR protocol, which we use for brain tumors, and especially in our case for glioblastoma? So we started with different MR protocols from 3 and 1.5 Tesla machines from 2 vendors and 3 centers, and there are more to come. We wanted to base our best MR protocol on, of course, technical improvements, the latest scientific literature, and we want to have a comparison of images by advanced readers. All in all, the aim is to make the protocol detailed and also, and this is important, adaptable to local differences, different scanner hardware, different acquisitions. Next slide, please. To give you an example of the literature which we considered in our decision-making, as an example here, the paper by Elixon, which is at the consensus conference, an international meeting of experts on this, in this field, and they came up with a minimum standard protocol for 1.5 and 3 Tesla MR, but also in a protocol for 3 Tesla protocol, and also a recommended protocol for 1.5 Tesla, not shown here. So this was taken into consideration. Next slide, please. To give you a short example of the very complex and extensive evaluations we did, is some parameters are shown here for 3D T1 weighted sequence. So some of the parameters that are shown here were finally decided in the best consensus by the technology improvement, some by the majority, and some by the literature. So at the end, we came up with a consensus best of protocol for, in this case, 3D T1 weighted sequence at Siemens Skyra. Next slide, please. So we did it for 8 sequences, and we proposed the best of compilation of protocols for these 8 sequences based on our physics principles, latest scientific literature, and clinical practice, and had the control data. This is going to be in the future more detailed. We will also compare the clinical images by advanced readers, which such started in detail, and assess the uses of consented imaging protocol within daily routine, and also analyze the effect of imaging parameters for AI processing. So thank you. I'm at the end of my talk, and I would like to hand on the mic to Roland Wiest.
Yeah, thank you very much, Andrea. You have already mentioned the importance of robustness of images, and this was also part of our project. And if I could have the first slide, please. Then you will see on top, that's the standardized protocol that has been mentioned before. So there are some basic recommendations on how to do glioma imaging based on the so-called Ellingson protocol. But if you take a look at the left side, this is a survey in more than 200 centers in Europe. So about the time and the acquisition modes that are carried out in these centers, then you see there is a huge difference among different European countries. And if you imagine, it's very important to have a strong definition about all these omic features that continue shape, appearance, the localization of the tumors, but also things that are related to the metabolism, then everything that is not aligned will impact on the quality. On the right hand, you can also see the different protocols. There are only slight differences in between these protocols in Switzerland, but also they will impact on the data quality. And if I may have the next slide. So what we did is we implemented all these protocols on one scanner. We had 2 persons that were repetitively examined, and we looked how these factors, these different factors like quality, blurring, then signal changes that are related to the hardware, to the noise, but also the reading quality was affected. And if I may have the next slide, please, then you see, well, in a perfect world, all these columns would look the same way, but you see there is, with these common protocols, still a huge difference in between the different persons that get examined, and these differences, they are related to image artifacts, they're related to contrasts, And they are also related to the overall diagnostic quality, as you can see on the right hand. And one thing that is really something that touches me very much is that younger colleagues rate the image better than those that have already a lot of expertise. So it's very important that experts see these images and that we try to improve them in the way with the new protocols that Andrea has mentioned. In the second part of the project, it's getting still a bit more complicated because we will also read the tumor images from different centers, and we will do the same ratings also with the new protocols that have been proposed. And we're happy to see if this improves the image quality for the expert reader a bit more. In the second part of my talk, I would then come, and maybe I could have the next slide here, a bit on factors that we can directly manipulate. That means we can give recommendations for the pre-processing so that the algorithms can do their proper work according to the recommendations that have been done in advance. And the community, and maybe you can have the next slide here, please, again. The community has still a debate about how these segmentations shall be carried out. There's an international activity, the so-called Brain Tumor Segmentation Challenge for the last 8 years. And the initial recommendations were that the different compartments of these tumors should be investigated separately. There's the edema, there is the central necrosis, there's the enhancing tumor that you can see in blue on the right image, and there's part of this non-enhancing tumor. But it turned out that these kind of separations are very hard to be segmented, not only by experts, but also by the algorithms. So there was a modification suggested that only consisted of 3 areas. That's the whole tumor, the tumor core, and the enhancing tumor. But still, these recommendations, they do not follow properly the biology that lays beyond this analysis, because tumor core is consisting here of unenhanced tumor and also of central necrotic parts. And that motivated us to have some discussions with experts in the field and to come out with a new definition of these compartments. And maybe I may have the next slide, and we have started to propose a more simplified composition that just consists of the two tumor areas that are really crucial for the outcome of the patient. That is the amount of the central signal abnormality, and we called it specifically a signal abnormality because it may have parts of necrosis, it may have parts of hemorrhage, and so there is not one signal, but there is one compartment. We also stick back to the definition of having the contrast enhancing tumor as one part and then uniting the non-enhancing part and the edema. That means the areas that extend over the enhancing part of the tumor into one compartment. And this will be the definition for the future work. Then may I have the next slide, please? The second step of the work is now related to these expert annotations. That means we will provide in these 40 patients that we receive from the different centers, first the manual segmentations, then we will apply different automatic segmentations, and we will see how these segmentations impact on differences in the radiomic feature extraction. We have started first with the pre-processing, and maybe I have the next slide here. And we just looked simply on the effects that are just related by some very simple things. That is the size of the image, the so-called cropping. That means you minimize the areas that do not contain significant information until a point where only the brain is covered. And you can see from the left of these tables, and this analysis has been done in more than 300 data sets from an international data collection, that there is just the effect related to the size of the image. And then if you use the same image sizes for the analysis and you just change one step in the normalization procedure, as you can see on the right side, then the effects of this one single line in the code manipulations, they do directly affect the quality of the analysis. Next point, please. And therefore, our first take-home message here is that we need the strong recommendations how the pre-processing needs to be done, and the changing of one parameter in the network may have a huge influence on the data quality. So to sum up, so first we need, of course, the standardization of the imaging protocols. This is ongoing work, including patients right now. We will come out with the standardized pre-processing and quality requirements for image cropping and normalization. We will have a look at the quality assessment of these glioma patients, and this is a part of the ongoing work package at the moment. And we are really happy to work. And with this, I would also like to hand over to Professor Mauricio Reyes, who's professor at the Biomedical Engineering Department at ARTORG. We have really good algorithms already developed in our team, in Mauricio teams, that we will use for the comparison right now.
Thank you very much, Professor Wiest, for the kind introduction, and welcome everyone to our webinar. It's a pleasure to be presenting this part of the SPHN imaging project. So as you have seen already, delineation is a very important area in radiomics. So it's the source of the radiomics or the information extraction. What you see here is an automated segmentation where the algorithm is extracting or delineating in a semantic manner the different parts or different sections of the structure of the disease as well. This is important. There's multiple sources of resources that goes into radiomics information extraction that can be also used for disease progression analysis and so on. Next slide, please. So in this area, the automation process has received quite a lot of attention, as Professor Wiest mentioned. The brain tumor segmentation challenge has been running for now 8 years. And what you see here is the state of the art. Now it's maybe a bit updated. This is a summary of the last years. But basically the main message is that we are getting closer to human performance, although we still see issues with robustness. And this, has also been discussed before, highlights the importance of normalization, the importance of also taking care of the imaging process. So we are getting closer to the performance of the human. So we can now, the potential is to automate this step such that we can use them on a batch mode where we can extract information at a larger scale and in a time-effective manner. So next slide, please. So as an update for the activities for our project here, what you see, this is the beta version of the software we've been working on. We call it Deep Bratumia. So Bratumia stands for Brain Tumor Image Analysis, and Deep Bratumia now is the deep learning version that so includes deep learning aspects into it. We have been very careful on the design to make it friendly to the user. We want the user to interact with the tool, to perform corrections, to be able to visualize the information, and also to extract then the information later for the next parts of the pipeline. So next slide. So as features of the tool, what the tool can offer. So it's deep learning based. So that means it's using state-of-the-art technologies. What we use here is we use a fusion approach, which means based on observations in the literature, is we take top methods worldwide, and then we perform a fusion approach to perform a final segmentation. Skull stripping refers to the task of removing the skull, which is important to enhance the performance of the segmentation of tumors, but it's also pretty useful when doing anonymization of the information. Since it is deep learning, it's running in few seconds. It's, as I said, it's designed for researchers and clinicians, and very important, it's compatible with the Biomed IT platform. It's all, we have actually refactored the complete code to make it completely Python enabled for the Biomed IT, and therefore it's ready to go for radiomic studies. Next slide, please. So our next steps, we are very soon ready to release the first alpha, a beta version to the scientific community. Our next steps is to then make it available also for macOS users and Unix users. We are planning for an integrated quality control and also an integrated AI-based sequence detector to really speed up the process of inputting and outputting the information. And of course, new visualization features in the sense of reporting capabilities. So I think that's from my side. I just want to finish by highlighting the very amazing work from Michael Miller. He's our main developer of the tool, and really my credits also goes to him for this very nice work. So that's from my side. I thank you for your attention, and I want to now introduce my colleague Adrien Depeursinge, who is going to talk about work package 3B and quantitative image analysis. Thank you.
Thanks very much, Mauricio, and good morning, everyone. So it's my great pleasure to share our efforts here from HES-SO Valais on quantitative image analysis and also machine learning. Next, please. So a first problem that we are tackling in this project is within Radiomics, we need to be able to analyze very local patterns that are also very directional, but can also appear at any local orientation. So one example here, if you consider this brain tumor, this glioblastoma tumor on the lower right part of the image with the red arrows, it's important to detect that there is a sharp boundary here between the outside and the inside. There is necrosis inside, but this sharp boundary will obviously appear at any local orientations. And so this is a challenge with regular deep learning models, because you need to relearn the detector for any local orientations. This is typically a huge waste of parameters here. So one first effort we did is to come up with a method where we can learn just one filter that can do all orientations. So next, please. So we started to test that with first a synthetic data set here. And what we see in the two graphs on the top right, on the bottom right, is that here we're measuring the performance of the approach while varying the number of training examples we show to the systems. And so what we can see is that our approach can learn much faster because there is much less free parameters in the network, because we don't need to relearn all these filters for all orientations. And in 3D, it's a lot of things to learn. And so on the right side, we started to test that also for classifying lung nodules in low dose CT from benign to malignant. And it seems that it's also important for medical images to adapt to these local patterns. Next, please. Another effort that we are working on is to join this image biomarker standardization initiative, which goal is really to standardize the definition and software implementation of these quantitative parameters. For instance, if there are two softwares and they are computing the same texture parameter, we really want to make sure that they're using the same mathematical definition, and also it's implemented the same in the software. So our role here was to write a new reference manual on convolutional filtering that we issued on June this year, and also a new benchmarking methodology for these different softwares. And we're also developing and hosting a submission website for all participants to upload their results and make sure they are achieving the consensus. Next slide. And finally, the third effort that I'd like to share is our platform, a web-based platform for radiomics research. So the goal here is really to facilitate and homogenize radiomics research in Switzerland. And the idea here is that if there is a clinical researcher that cannot code, but has excellent hypotheses that he wants to test. For instance, he wants to test if the T2 image can predict overall survival of a glioblastoma. He can use this platform, which is user-friendly. So what we did here is to use an open source software, which is KEOPS, to manage image collections. So these are from the developers of Zurich. And then we plugged our image analysis platform to do feature extraction, so quantitative image analysis, and also machine learning. So you can test the platform with anonymized data using the link that is shown here. And I will now also just do a little demo under the form of a video of this prototype platform. So next, please. So here you can arrive in this platform following the link that was shown before. You can log in into the platform, then you arrive in this KEOPS platform here where you can really manage your image collection. So this is something we don't develop, but we just use this open source software. You can really check that your images are really the ones you want to analyze and scroll through the images. You can also look at the different region of interest. For instance, here we have pulmonary tumors with lymphangitis. So we have the GTVT, which is the main tumor, and so region of lymphangitis. So when this is all ready, you can switch over to our platform with the same login. You will have exactly the same view as the data in KEOPS. And here you can look at the different cohorts that you have, and you can start to do batch feature extractions for all your cases in the cohorts. For instance, for this lymphangitic dataset with 91 series, you can start to extract features. You can choose if you want to do intensity features, shape, or texture features. We use Radiomics software, which is very standard. And also we have our own features like Read3D, for instance, which is also this locally rotation invariant. Then you can extract features, takes a bit of time. And here you can download your features either to use externally with another software, such as statistical software, for instance. If you do so, you will get a zip file with a collection of spreadsheets for each image modality and region of interest, basically. Then you can also use our platform for machine learning. And here you can just click on this button to train a model. And here, if you do that, you can choose between classification or survival approach. If you do classification, then we have various algorithms that are available. And then you need to also upload labels. So here we will test with just classifying between lymphangitic and non-lymphangitic patients. And if you do that, then you arrive to this page where you have a summary of what you did. So we did here classification, using logistic regression. We use a 5-fold certified cross-validation, CT, texture feature, etc. And then you have your model metrics with AUC, conclusion matrix, etc. And you can decide to stop here, or you can go back to test another model or to test new features to really converge to one model that is interesting. So this is all for my part. Thanks a lot.
Well, Adrien, thanks a lot for giving the last presentation part. This brings us to the end of the presentation. We hope that we were able to give you a good overview of what we tried to achieve in the Imagine SPHN driver project. We hope to motivate you, to interest you in this type of research. At the end, always, and I think this is very important in particular for this project, it's a team effort. We have a large number of sites. We have a large number of people who dedicate themselves, who contribute. So many thanks to the people who have contributed today in this presentation, but also in particular to all people who are contributing in the background, doing a lot of work. And we had a very good and promising start. We are on a good way, and we hope we can keep this spirit for the next years to drive the research in radiomics, to integrate that into the SPHN, hopefully despite the issues we are also facing with the COVID issue. So thanks a lot, and we are happy to take your questions.